Let's talk about that, because again, you're talking about influencing this very interesting sense of a biological clock, which is not exactly the theoretical physicist's clock, right? Really quite different. You talk about influencing it epigenetically with external influences. Can you tell me about one of these experiments with this slime mold here that you did? Because I think it really illustrates the point of how this information theory of aging works.
It's a bit of a story. I started off studying the epigenome in yeast cells at MIT, and I landed at the right place at the right time, speaking of serendipity. And under the tutelage of Leonard Guarenti, we started trying to understand why don't yeast cells get old? Why do yeast cells get old? Why are they not immortal? And these are the same organisms that you use to make beer and bread, so you just grow them on plates. And we figured out why they get old. It turns out that they have genetic instability. They get broken DNA, same way we do. Every cell gets broken DNA every day. A few chromosomes break every day. But that then distracts the epigenetic control systems, because the epigenetic control systems also help repair DNA. And I believe that that's important, because you want to control your epigenome in response to DNA damage. So why not have the same proteins control both things? And these proteins, we gave them a name. We called them Sirtuins. There are 7 in our body, 5 in yeast. And they don't just repair DNA, they protect against aging. So when you put more Sirtuin copies of the Sirtuin genes in yeast, they live longer. Turns out that's also true for fruit flies and worms and mice, yeah. But then the question was, well, are these epigenetic changes really causing aging, or is it just something that happens, and what we'd call an epiphenomenon, an association? And in science, as you know, you want to break something and see what happens, right? And so what we thought we'd do is we'll go in and break some chromosomes. And if we're right, then the yeast cells should get older, and they did. They aged rapidly. And then in 2007, I said to a postdoc in the lab, Philip Oberdorfer, how about we do this in a mouse? And he said, you're crazy. It'll kill the mouse, or it'll get cancer. And I said, okay, but just do it. And he was pretty stubborn. And what I said to him was, all you have to do is prove me wrong, and then you can do something else. Just prove that this won't work. And actually, 2 years later, he came into my office and said, I think it might be right. But he was really skeptical. So what we did, and why it's relevant to this organism, you can see here, is this slime mold, Polycephalum physarum, makes a very special enzyme that's part of its immune system. And when you purify that enzyme, or take the gene for that enzyme and put it into a human or a mouse cell, it will cut the chromosomes very rarely, which is good. You don't want to cut the chromosome to pieces. It'll kill the cell. So it cuts the human genome in roughly 20 or so places. And the other good thing about it is it makes staggered cuts in the DNA. So it cuts the DNA ladder over here and then over here. So you end up with what are called sticky ends. The bases of the DNA come apart like a zipper, and the cell can just find those ends and put it back together. So you don't get a mutation the way we did it. So now we could test the hypothesis. If you distract those Sirtuin protective enzymes with cutting of chromosomes, what happens? And the mouse could have died, nothing could have happened, could have gotten cancer, could have gotten aging. And I remember I was in Australia, and I had one of the first iPhones, and I got a text saying, the mice are sick. And I said, okay, send me a picture of the mice, I want to see. And I saw a picture of a mouse, and I said, that's not a sick mouse, that's an old mouse. How old is it? She said, it's 2 months old. I mean, let me guess, that's the one that had this enzyme in it.
So you accelerated the aging process.
We caused aging in an animal, and we published this last year in the journal Cell, and it was 43 scientists. It took us 13 years of work. It was a lot of work to convince the scientific community that we caused aging and not just made a mouse get old or look old. But by every aspect, it was old. It had an older epigenetic clock. We can measure DNA methylation changes, they're called. We can track that. It was older, but it had all the signs of aging. And we can dial up aging as fast or slow as we want in those mice. We can make them age in 2 months or in 16 months, but we definitely caused aging. And this was the first evidence that the information theory of aging was true for a living organism as complex as a mouse.
Well, see, I'm a little confused because I thought you were curing aging and you're causing aging.
Well, we did that to test the theory, but once you can cause something, we believe you can take it away, and that was our next project.
So are you saying that the Sirtuins are somehow, you're honing in on as a fundamental vehicle for both the accelerating aging process as well as the reversal of the aging process?
Yes, 100%. We have a lot of evidence that that's true, that these Sirtuins protect you, but if you distract them with other problems, if you smoke, if you eat a lot of processed meats or other things, go in the sun, you will distract them and they won't be protecting your body. They'll be doing other things. So what you want to do is
encourage them.
Encourage them. You can simulate damage without causing damage. You can fast. You can go for a run. You can jump in a sauna. These are things that turn on the Sirtuins. It's called hormesis. What doesn't kill you makes you stronger, but it cannot be damage that's so overwhelming that it's a net negative. You want to be a net positive. And the Sirtuins get less active as you get older for a variety of reasons, and we can either chemically or lifestyle-wise raise their activity back up again so they can simultaneously, or there's enough of their activity that you can repair the broken chromosomes and maintain the epigenome in a youthful state. And we have some evidence now that in this rejuvenation state, the frog experiment and also our rejuvenation of the animal, that the Sirtuins are involved, and what's happening is that they're going back to where they should be rather than being lost somewhere because of getting distracted by DNA
So
breakage.
you're luring them back to do their job again, to
Yes.
keep the DNA.
Yes, and so here's the biggest question in my lab, and arguably one of the biggest in biology. How do they know where to go back to? Where is that information? And we're looking for that.